Hello, welcome to Low Tech Whiteboard Learning. Today we're going to talk about membranes. And why are membranes important? You might be wondering, why is she talking about that today? And is that really important to me? But membranes are really important, and not only because they keep certain things separate in our body that are important to keep separate, but also because they help form gradients. So the subtitle for this would be Gradients, Why You Care. So let's talk about membranes. So membranes do a lot of things, right? They separate certain parts of your body from other parts of their body. So they're big membranes. So one would be the blood-brain barrier. So your brain is here and the blood goes by. And as the blood is going by, that barrier keeps certain things out but lets certain things in, like energy and oxygen. And other ones would be your peritoneal membrane, and your capillaries are sort of membranes, and you can have problems with your membranes. So one of the things you may remember from biology is that we have a phospholipid bilayer, and so one of the things that can happen is in this phospholipid bilayer membrane that your cells are made up all the way around, is if a bad guy, an oxidative, like an oxidative stressor, comes by, a free radical, like hydrogen peroxide, can come in there and break this apart, and then your cell membrane is split open. So that's how antioxidants work. So membranes are super important there, or else the cell just can't function anymore. Membranes also, when they help us with gradients, make things work, like electrical signals from nerves and the way that your kidney functions. So we're going to talk a lot about that today. So we definitely think membranes are important, or at least I'm telling you to think that membranes are important. So what is it about membranes that give them so much power? Well, defining a membrane, it's one thing separated from another, right? And there's something in between. It could be synthetic or something in your body. The membrane itself is important if it is permeable to certain things coming through or impermeable. And there's very few impermeable membranes in your body that are that important, but the grossly big ones are. So maybe like the pericardium is a relatively impermeable membrane, and we were talking about some of the other ones, like the aorta is impermeable as it comes out of the heart. Things don't move through that. That's because it's thickness. The cellular membranes, so the ones that are very small, microscopic membranes, when we talk about those, usually they are permeable. And they are either semi-permeable or selectively permeable. And it really doesn't really matter that we necessarily know the difference, but it is kind of fun to learn. So let's talk about why something might be semi-permeable, which means it lets things through some if you're the right kind of molecule. It's very selective, right? It's not semi. That's confusing. So semi-permeable membranes let things go through based on some sort of criteria. And that criteria might be size. So maybe the little ones can go through, but the big ones can't go through. Or another membrane might have a big size, and so big guys can get through and so come to the other side. Whereas other semi-permeable membranes may decide that their pores are going to be charge related. So if this pore has a negative charge all the way around it, then only maybe positive things will get through. Because like magnets, the negative would get rebuffed. So charge is another way that semi-permeable membranes keep things from coming through. A selectively permeable membrane has a specific active way of letting certain things through. So that might be a channel or a pore that actively grabs something and pushes it through. So those are actively allowing certain things through by having pores. And those are what's really important for some of our cellular function. So let's talk about the most basic cellular function, that help the gradient. That's the most important to me, and that is the sodium and potassium gradient. So in our cells, we talked about this before, in your cells you have a higher concentration of potassium than you do sodium. And outside the cells, in the interstitial fluid, there's a higher amount of sodium. And so that's the interstitial, and that's the vascular, and that's the, obviously the cell. So potassium is kept low, maybe at 4 out here. Potassium is really high here, like 150. And that's the same for sodium. Sodium is about 150, and in here is really low, like 6 or 7. So how does the body do this? Well, the cell membrane is not naturally permeable to sodium or potassium. And so in order to make this gradient, there has to be a pump. And there is a pump here that when a sodium comes by, meaning probably that there's a little bit too much sodium in the whole cell, those sodiums load up, 3 of them, and they get pumped out. And then potassiums will come by and they'll get pumped in. And this is the 3 sodium, 2 potassium, ATP ACE pump. And why is it ATP ACE? Well, because it needs energy for this to happen. So this isn't a natural process. This is an energy propagated activity. This is really important because if we don't have this gradient, we don't have natural amounts of sodium that are high here and potassium here. We can't have that naturally without the active pump. We also can't keep potassium away from some cells that are very sensitive to potassium, like your heart. And so potassium has to be kept away. Another way this is done is inside the cell in the sarcoplasmic reticulum. There are calcium deposits, and calcium is kept away from the rest of the cell. And so if calcium leaks out, the cell doesn't function very well. Now, the important thing for this in many ways is not only that we have this gradient, but what happens when you're in shock. When you're in shock, you don't get much blood flow, you stop bringing oxygen into the cell, and you stop bringing nutrients into the cell. So the cell suddenly doesn't make energy, and we lose this. If you don't have energy for this, you can't maintain these gradients. And so the cell membranes don't keep calcium where they're supposed to be and can't function to restore sodium and potassium the way they're supposed to. So that is one of the effects of shock. And so potassium could come out and hurt the cell. Sodium can rush in. The other thing that happens that's really important about this is the gradient itself is energy. So if I have a lot of potassium on this side of, potassium here and sodium here, I can pair these things up to each other. And one of the more interesting ways that happens is with phosphorus and potassium in diabetics. So some of you may know that we're really, really worried about low phosphorus and low potassium when we start insulin therapy. Well, why is that? Well, one of the reasons why is that, we keep our potassium in a tight range at like 4, and phosphorus is a relatively tight range, but we have our potassium and phosphorus, there's pumps here. So when we have insulin around, insulin binds here and drags a sugar into the cell because we're trying to get sugar into the cell. And because we are driving those things in, there's actually a cofactor, and it will drag in a potassium and drag in a phosphorus. And so we will, because of insulin and this pump getting sugar into the cell, we actually will have a lowering of this and an increase into the cell. The other thing that can happen is you can use the sodium gradient there. And the sodium gradient, if you, let's say, I have all of a sudden I put in a salt here. So say my pet eats salt, and suddenly there's so much sodium around in here. Now, water, we didn't really talk about this, but water is pretty okay going in and out of this. It doesn't have any problem getting through this semi-permeable membrane. It goes freely in both directions. And so usually the water kind of stays where it is. It likes to be around the sodium. It likes to be in the cell. But suddenly I put extra sodium on this side. Water in the cell says, hey, we got to dilute that out. We can't have so much high sodium out there. We're attracted to that sodium. And suddenly water goes through the cell membrane. And then if that happens a lot, and all the water says, okay, we got to make this in cell water concentration the same as out here, then all this water has to go out here, and that cell will actually shrink. And so that's what happens with hypernatremia. If somebody eats salt or goes in the salt water and you get salt into the system very quickly, water rushes out of the cells to dilute that sodium and shrinks the cells. And it's not that big a deal if it's your muscle cell, but if it's your brain cell and this is your skull, that's going to cause this space to get opened up as the cell pulls away, as the brain cells pull away from the skull, and that might fill with blood. Now the opposite is true if you have too little sodium in the blood. Like suddenly you drink fresh water. So let's say we have a Labrador who goes crazy and just drinks out of the hose for like an hour, and all of a sudden there's just a ton of water out here with your normal amount of sodium. Extra water is going to say, hey, there's some attractive space over here because I've got to make the concentration of salt out here and here relative to water the same, and the water rushes in and the cell bursts or it gets big. And what if you do that in the skull? Well, that causes brain swelling. So the gradients are important, but also the membranes are important. And so we'll talk about that a little bit more when we talk about hypernatremia and when we talk about shock. I didn't really even talk about nerves, how nerves and muscle fibers work by the release of calcium and potassium. But this is a good place to start so that you understand that certain membranes allow certain things through, and it's an active process if you want to work against that. And one of the other diseases we can talk about is when we're checking things like uroabdomen and septic peritonitis, where molecules can get stuck on one side of a membrane or another. So that's a good place to start. And I hope that wasn't too confusing for you. And we'll see how it goes for the next installation of membranes and gradients.